Hello, all you biology folks. I wanted to show you a virtual lab that's pretty short and it does a good job explaining and showing the differences between a hypotonic solution. And remembering the rhyme, hypo means there's low solutes outside. And the other way to help you remember is hypotonic solutions cause the cell to gain water like a big hippo. So hypo, hippo. I know that's silly, but it works. So a hypotonic solution means that the cell has low amount of solute outside, but a higher amount of water outside. We're also going to look at an isotonic solution, meaning that the inside and outside solutions are the same. So you have the same amount of water, and so water is going to enter and water is going to leave at the same rates. And then we're also going to look at a hypertonic solution. And remember, again, it rhymes. A hyper has a higher number of solutes, but that also means that there's a lower amount of water, and that will cause water to leave the cell and diffuse outside. So as I show you each of these cells, I want you to fill in the chart that's in our Google Slides. You want to look at whether or not the cell gets bigger, the cells, in other words, cells gaining water, whether or not the cell stays the same, meaning that the cell gains water and loses water at the same rate, or whether or not the cell gets smaller, in which case that means water is losing. The cell is losing water. All right, so we got three cells here. We've got a red blood cell, and that's a typical, animal cell. It does not have a cell wall. And then the allodia is a plant cell, and so it does have a cell wall. And then we've got a paramecium. So let's take a look. So I'm going to drag the red blood cell in a hypotonic solution. And remember, you're going to record what happens. Okay? So did the cell get bigger? Did the cell stay the same? And notice the arrows, did water enter or leave the cell? All right, I'm going to put it in the isotonic. All right, and notice I'll do it from the normal. And notice, did it stay the same? Did it get bigger? Did it lose water? Notice the arrow should tell you what's happening there. Water going in, water exiting. All right, and then I'm going to put it in the hypertonic. Make your prediction in your mind. Remember, hypertonic means there's higher solutes, lower water on the outside. Okay, so did the cell gain water, lose water, or stay the same? Okay, so I'm going to do that with the allodia as well. Now, one quick reminder, plant cells have a big vacuole that is the big storehouse of water. So you'll notice the vacuole changes as well. So let's go ahead and look at hypotonic. Show that again. All right, so describe what happens. All right, I'm going to put it in isotonic. And then I'm going to put it in a hypertonic. Watch what happens to the vacuole and the cell. Now, yellow spaces are just empty spaces from the cell wall. That's not a good thing. The cell has gotten smaller, but the cell wall has maintained its shape. That can do some damage to the plant. All right, let's do the paramecium. All right, that's hypotonic. And let's show you what it looked like normally. And then let's do a hypertonic. Okay. Now, normally paramecium is pond water. Pond water is fresh water. So I'm going to show you a short video in class about what paramecium can do to fight off the pond. Remember, pond water is, because it's fresh water, it's going to be most similar to hypotonic. And so normally it would be gaining water, but is there a way that cells can kind of fight this? And so I'm going to show you a little video in class where some single cell organisms have this ability. All right, so that will help us just demo our lab.